The problem that we have is that clearly life must have evolved after these turbulent times, about 4 billion years ago, and older than 3.5 billion years ago, but we don't have any sedimentary rocks from this period. And if we don't have any sedimentary rocks, we are faced with a problem because we therefore cannot know the pH or how acidic the primitive ocean was. We have no idea of the composition of the primitive atmosphere. We do not know what was the temperature of the Earth. We have a big question mark, and let me warn you that this question mark will appear over and over during the rest of the lecture. We do not know how life appeared on Earth. Nevertheless, we have a very reasonable hypothesis that was proposed in 1924 in the Soviet Union, and then developed in an extraordinary way a few years later, in 1936, by Alexander Ivanovich Oparin, a friend, a professor, a colleague that I had the chance to interact with, who proposed the idea that first life had been heterotrophic. In other words, it was finding its sources of nourishment from organic molecules that had synthesized and had accumulated on the primitive Earth prior to the emergence of the first microbes. And Oparin's ideas were absolutely extraordinary because what he did basically was he showed an absolute disregard for the frontier separating different scientific disciplines. He was trained as a biochemist, but he decided to get up and look what was going on among the astronomers, among the geophysicists, among the planetologists, and came to the conclusion that the primitive Earth had an atmosphere that was highly reducing. Highly reducing meaning that it not only had no oxygen, no free oxygen, but also that it was rich in methane, hydrogen, ammonia, and a number of very reduced of compounds rich in hydrogen. Oparin proposed that the solar radiation, the heat from geological activity, and so on, led to the synthesis of organic compounds, that these compounds were dissolved by rain into the ocean and formed the primitive soup, el minestrone primitivo, la sopa primitiva, vichyssoise, as you wish, and that in these oceans rich in organic molecules, the interaction between the different molecules led to droplets, the formation of droplets rich in organic molecules that began interacting among them, and from here these droplets, which he termed quasorbates, evolution led eventually to the first bacteria. They were heterotrophic because they were nourishing themselves from the compounds, organic compounds already available. They were anaerobic because they had appeared in an atmosphere, an environment that had no free oxygen. And they were prokaryotes because prokaryotes are the oldest forms of life. Now, if you look at this picture, you will see that there is no mention of nucleic acids for a very simple reason. When the book was published, James Watson was going to grammar school. The idea of double helix DNA as a depository of genetic material was not part of biology. Nevertheless, the theory is what we call in epistemology an open theory. It could accommodate a number of later discoveries without destroying the overall essence of the scheme that Oparin proposed. Now, do we have any evidence that indeed there was this synthesis of organic compounds? Yes, and the answer comes as a gift from the sky into Australia. In September 28, 1969, a meteorite that we call now the Murchison meteorite because it fell in Murchison, came into the Earth, was quickly collected by a number of Australian farmers and scientists, and don't ask me why, but most of the meteorite ended up in the United States at NASA. Now, the very nice thing about NASA is that if you have a good project, you can actually receive parts of the meteorite and you can analyze it. And the most wonderful thing about this meteorite is that it turned out to be as old as the Earth itself. It's 4.6 billion years old. In other words, when this meteorite formed, the Earth itself was condensing. So here we have a sort of geochemical fossil of the conditions that were taking place on the Earth, on the solar system when the Earth was being formed. And the chemical analysis of this meteorite, which is nothing extraordinary, it's the same kind of analysis that you will do if you want to be tested for cholesterol or diabetes or whatever, and you go to the hospital and they take some blood or some urine and they do a number of analytical procedures. It's exactly the same procedures that we use, but but instead of urine or, well, we have urea here, but instead of urine or blood or saliva, what we actually analyze is part of the meteorite, and we discover hydrocarbons, amino acids, around 80 different amino acids, purines and pyrimidines, which are the components of nucleic acids. We discover urea, sugar derivatives, and we know that those sugar derivatives come from the transformation of deoxyribose and ribose, which are the sugars that we found in nucleic acids. We discover lipidic components, which are the what forms membranes. We discover alcohols, and not the ones that you would like to see, and carboxylic acids. So quite clearly, the meteorite is telling us that when the Earth was being formed, there were a number of chemical processes going on in the solar system. They were forming organic compounds, but not any kind of organic compounds, but especially the ones we associate with living entities. The same biochemical monomers that we see in living beings. So this clearly reinforces the idea that a similar process was in fact taking place on our own planet. Now, this is one way of confirming or studying the possibility of organic compounds in the primitive Earth. The other one is to simulate under laboratory conditions how they may have taken place. And the first to do so in a very successful way was my friend, my colleague, my professor, Stanley Miller, who, when he was graduate student in the University of Chicago, working with Harold Urey, one of those extraordinary scientists, one of those extraordinary characters that Americans have all the right to feel extremely proud of. Stanley Miller decided, after he had been at the school for only a few weeks, that he wanted to do his PhD thesis trying to simulate the chemistry of the primitive Earth. And he actually designed a very simple experiment with these two flasks. In the upper flask, he had methane, ammonia, and hydrogen. And here in the lower flask, he had boiling water. The water, as it boiled, came up here as vapor and mixed with the other gases. This was subject to the action of electric discharges for 1 week. And you can see how, after, in fact, a few hours, the chemical composition of the inside has been transformed. And Stanley Miller was able to show, when he was only 22 years old, that indeed you can simulate the primitive Earth and synthesize without any major problem organic compounds such as amino acids. The paper was published in 1953, and in fact, if one looks at back in time, 1953 was what we can call an annus mirabilis by all accounts. Why? Because it was the year in which Watson and Crick, two young, bold, dashing scientists, they did not know sufficient chemistry, did not know enough physics, but were very clever and had the right intuition, came out with the idea of the DNA molecule as a double helix. It was the year in which Sanger and Thomson say that insulin is a protein in which the amino acids are not located randomly, but follow a strict order, something that we diabetics are very happy with. And finally, is the year in which Stanley Miller demonstrated that under prebiotic conditions, you can synthesize organic compounds. The vintage of 1953 was not very good for wines in France. I discovered that. But it was extraordinary for biology. It was extraordinary for science, because these three discoveries made by young, bold people who had the willingness to try and go against the tide actually demonstrated that you can understand in a very logical way some of the components of living entities, some of the things that led to the origin and evolution of life. Now, the experiment by Stanley Miller actually opened up the laboratory study of prebiotic chemistry. And I think that nowadays it's fair to say that we can imagine the primitive Earth as a place in which under, for instance, hydrothermal vents, you can have the synthesis of organic compounds. We can have electric discharges, and this can lead to the formation of organic molecules, as Miller demonstrated. We can have the same when we simulate the conditions near volcanoes. And of course, we can picture without any problem the infall of meteorites or comets rich in organic molecules. If you look at the articles and the books published, mostly by my American colleagues, on the formation of the primitive soup, you will see that there's a tremendous discussion of which of these sources was more important in cooking the soup. Some people will argue that it was hydrothermal vents, others will argue it was electric discharges, some will say no, what came from comets and meteorites is what led to the accumulation of amino acids, nucleobases, sugar, lipids, oligomers of these biochemical compounds. If you look at the discussion, it's very interesting because I think it's a very American discussion. And the question becomes, have too many cooks spoiled the soup? And I think the answer is no. If you come from cultures that have developed very sophisticated cuisine, like the Mediterranean people, like India, like China, like Mexico, or like Australians, you quickly realize that if it works, it's edible. You never despise any single source of organics. Or in Australia, I should say, if it jumps, it's edible. You never despise any single source of organic material. So some of us have the impression that one shouldn't think of a single source of organic compounds for the primitive earth, but rather have a very eclectic view in which different processes led to the accumulation of different amounts of organic compounds. The more organic molecules you have, much the better. And of course, we have not solved the issue of the origins of life, but if I had to do a balance, if I had to analyze the contribution done by my friends working in prebiotic chemistry, I think it's fair to say that although we do not know how life appeared on Earth, all the evidence that we have suggests that prior to the origins of life, the Earth was already endowed with a very wide array of organic compounds of biochemical significance, that there were many organic and inorganic catalysts, that there were purines and pyrimidines, which means there's the potential for nucleic acid replication, and finally that there were also membrane-forming compounds. Once again, let me underline, we do not know how life appeared on Earth, but what I am listing here are in fact properties that we normally associate with living entities. Organic composition, catalyst, replication, and isolation and individuality by membranes. So I think that in spite of the many uncertainties that we have regarding the origins of life, what we know of organic chemistry, of prebiotic simulations of the primitive Earth, strongly suggests that we are working on the right path. Now, what happens if we try to put all these things together? Well, Jack Szostak from Harvard University did precisely that, and in an extraordinary set of papers that the most significant one, I think, was the one published last year in Nature, he demonstrated that you can come out with droplets made of lipids. You can put inside the droplets DNA, which happens to be very stable, and it's a very good reason for choosing it. And then outside you can put the nucleotides, and the nucleotides will go across the membrane, very much to the surprise of everyone. Theory did not predict that. They can actually polymerize reading the template, reading the single-stranded DNA you have on the inside. And as the droplet grows more and more and more because of the accumulation of organic compounds, it will break up into different droplets. It's not a living system at all. But here we have the possibility of something that looks like replication of nucleic acids inside a membrane, and it's strongly reminiscent of what happened in cells. So this probably implies that we are actually along the right way, working on the right direction. Now, what we would really like to know is how did, in this primitive soup, develop what Francis Crick once called the central dogma of molecular biology. We know that nowadays DNA can replicate, stores genetic information, and replicates thanks to proteins. We know that proteins do not replicate but are endowed with catalytic activity. And for many, many years, this became the chicken and egg problem in origins of life. What came first? Proteins that can catalyze or DNA that can replicate. And I have to say that the discussion went beyond laboratories, went beyond libraries and meetings and so on, and became a very political and ideological discussion. It's very interesting to read the papers that were published in the 60s and 70s, and to see that some of my more leftist colleagues were inclined to favor the idea of proteins emerging first, and they will say, because Engels says in Dialectics of Nature that a protein precedes is the central basis of life. And some of my most conservative colleagues will say, no, DNA came first, because it's like the general directing the army of molecules inside the cells. It's like the administrator of a big company. And it's fascinating to see how our intellectual, social, political prejudices can bias our understanding of biological processes. Well, one of the most amazing statements I have seen of how political the discussion came about, in 1961, Hermann Müller, a very distinguished, extraordinary good American geneticist, claimed that Oparin's refusal of acknowledging that DNA had appeared before proteins was part of a how did he phrase it? Was part of a Soviet conspiracy to run against the development of genetics in a worldwide level. The discussion became very intense. It became truly illogical in many ways, but the question remains, what came first, DNA or proteins? Well, the answer probably is that what came first is RNA. RNA, for many, many years, had been the ugly duckling of molecular biology. It had been just the go-between, il postino, as they will say in Italiano, el intermediario in Spanish. But RNA is actually a very peculiar nucleic acid. What does it mean to be RNA? Well, all of you remember that famous remark, the scene of the balcony of Romeo and Juliet, where are you, Capuleto and Mama Montesco, or other way. I always get mixed up with the families in Verona. But what's in a name for the case of RNA? RNA is a nucleic acid with bases attached to a ribose phosphate backbone. And this backbone has ribose, which is a sugar, OS is for sugars, and rib are the initials for the Rockefeller Institute of Biochemistry. Which means that at the intimacy of our cells, we carry the name of one of the hardest representatives of hardcore capitalism, Rockefeller. Regardless of that, what is extraordinary is the flexibility and the richness of special conformations that you can have of RNA. And this led in the late 60s to proposals by three different extraordinary scientists, by Carl Woese in 1967, by Leslie Orgel in 1968, and Francis Crick in 1968, to the idea that there had been a very early stage in life during which RNA had been the central molecule in biological processes. This was done in a time in which there was no evidence at all of the catalytic activity of RNA. But this gentleman had an extraordinary intuition of the many roles of RNA and proposed that RNA appear before proteins and DNA. And let me underline the word intuition, because normally we associate men with rationality and women with intuition. Intuition is sort of despised as a secondary way of thinking. I still have to meet a scientist, a good scholar, who lacks good intuition. Intuition was something that was abundant in the proposal made by these three gentlemen, and they were the first to propose the idea of an RNA world, a world in which catalytic activity was performed by RNA, and RNA was, in fact, the molecule that was also storing genetic information. And 20 years later on, their intuition was demonstrated to be true. We know now that RNA molecules can store genetic information, like DNA, like within the polio virus, in the influenza virus, in the HIV virus. And we know also that RNA can catalyze chemical reactions like proteins. And this led to the idea that once there was an RNA world in which a replicative RNA was encoding for a catalytic RNA that was helping to replicate catalytic RNA. And there was a wonderful paper published in February this year in Science by Jerry Joyce from the Scripps Research Institute, demonstrating that indeed you can have an RNA that self-replicates and goes and changes and evolves without any major problem. We have very good proof of the idea that there was once in the evolution of life an RNA world. So I think it's fair to say that we know how to cook a primitive soup. We really don't have any problem understanding the accumulation of organic compounds of the primitive Earth. We do not know, and let me emphasize this, we do not know how to get from the primitive soup into the RNA world. Perhaps they were intermediates, perhaps there were different kinds of genetic polymers that we know nothing about. But the interaction of RNA with amino acids clearly shows that protein biosynthesis evolved in an RNA world. We know that DNA was selected for because it stabilizes in a wonderful way genetic information. And once you have DNA, RNA, and proteins, you can have this extraordinary diversification of life. We can reconstruct most of the major steps that led to life and diversification of life on Earth. But furthermore, we can put that in a wider context. We actually recognize that life appeared very quickly on Earth, and the emergence and evolution of life eventually led to cyanobacteria. Cyanobacteria liberate oxygen as a waste product. This led to a major change in the chemical characteristics, in the physical characteristics of our planet. As oxygen began to accumulate, this led to the emergence of eukaryotes, cells that are the outcome of processes of endosymbiosis, and that eventually led to animal, plants, fungi, and a wide number of protists. And the extraordinary diversity that we see of life today is, in fact, the result of the co-evolution between the planet and the biosphere. The biosphere has not been the passive receptor of selection pressures put by the environment, but the biosphere has also actively changed the earth, the waters, the land, the soil of the planet that we live on. And all this, of course, comes from the inheritance of Charles Darwin. We clearly have to cherish the wonderful idea behind, the wonderful thoughts, the wonderful framework provided by the idea of natural selection and evolutionary theory. And the British actually celebrate Darwin in many different ways. For instance, they have a 10-pound bill with Charles Darwin picture, and we are slightly less materialistic, but nevertheless, we are happy to cherish the inheritance of Charles Darwin, and we even have a tequila that's called Evolution. Thank you very much.
Wonderful. Thank you.